All right, another protein class that we're going to look at is the immunoglobulins, which we looked at really briefly in that we know how to use them and we know a little bit about their structure. All right, I'm going to take a day and a little bit to teach you immunology, of which we have an entire semester. Okay, so I'm going to just give you a very, very brief overview. Did anybody take Dr. Christmas in immunology yet? If you haven't, it's a wonderful class, and you spend a whole lot more time investigating this. I'm just going to give you the brief overview. All right, immunoglobulins, part of the immune response to invaders to our body. Okay, so what the immune response is supposed to do, it is supposed to find the things that are not supposed to be there, things that are misshapen relatively. Okay. And so if something foreign comes into your body, it's a three-dimensional shape that's not necessarily supposed to be there. Also, if something within your body misforms, you're going to create something that is misshapen that can also be, quote-unquote, foreign, and then the immune response can be elicited to that. There are several kinds of immune response. There's a cellular component, which we'll take a look at it, and a humoral component, things that are in the blood, of which the immunoglobulins are one of the main components in that. The immunoglobulin, or Ig, okay, have different classes. The biggest class, or the most representative class in your blood system, is the IgGs. So sometimes we'll talk about IgGs as being what's going on. And so if you take a look at that, the structure of a antibody is kind of Y-like, and the crystal structure actually looks very much like a Y. And what you have is, you have two identical hands called the FAB region, the antibody regions, the binding regions, the ones that bind to the invader. We call that already an antigen, okay? And the antibody will bind to the antigen at what on the antigen? What do we call it? The place it actually bound. The epitope, okay? And I can grab onto it. The other part is the feet, okay? The feet are what makes the different classes of antibody. Most of them are the G version, and so what's called the FC region is what will tell the body what to do when it binds to something. Okay, and so there are different classes and different things that happen. The mass of an IgG is about 150 kilodaltons, about 150,000 grams per mole. And I can break this with a digestive enzyme called papain, and we'll break it into 3 fragments of about 50 kilodaltons each. What I will actually break it down will be basically there, and you get 1, 2, 3 regions when it gets broken down. The FAB binding site will bind to antigens with the same affinity as the whole IgG, but it can't what's called cross-link, meaning that a fully prepared antibody can bind to one antigen and to the other antigen and hold 2 of them together and cross-link them. If you break it apart, only one binds and the other one can bind, but they're not connected together. Okay. Immunity is brought about by white cells, okay, and the very basic definitions of these. The leukocytes. 2 types that we're going to look at, macrophages and lymphocytes. And then we're going to look at the cellular immunity and the humoral. The humoral has in them the antibodies or immunoglobulins and then beta cells. Okay? The cellular will use what are called lymphocytes, and particularly the T lymphocytes. You may have heard them as T cells. Okay, and we're going to talk about 2 kinds of T cells, the cytotoxic or killer T cells and the helper T cells. And we're going to talk about the proteins that function in these cells to cause immunity. Okay, we've covered this a little bit. The antigen is anything that gets bound or elicits an immune response. The antigen determinant is another name for epitope, is the actual physical place on the antigen in which the binding actually occurs. And then there's something called a hapten, okay? And we actually talked about this a little bit, but not articulated it. Sometimes you want to make an antibody to something that's relatively small. How do we make an antibody? What do? We take what we want to make an antibody to and do what with it? Inject it into a rat or a rabbit or something that has an immune response. Here's the problem. If the entity that you want to make an antibody to is too small, the body of the animal can filter it out. And it doesn't stay in the bloodstream long enough to build an antigenic response. And so what we do is we have to sometimes take our antigen and connect it to something that's big so it doesn't get filtered out. If I have to connect the thing I want to make an antibody to something, a carrier, that's called a hapten. And then that gets injected. Okay? You never know if your thing's not going to get filtered out, so you try it once. If you get no response, then you connect it to a carrier molecule, and then you inject it. When we actually break it into 3 parts, if we actually collect this part right here, it readily crystallizes. It makes a wonderful crystal structure. We can get a crystal structure from it, but it doesn't bind to anything. This is what, and the structure of this will tell the body what to do with whatever is bound. The antibody structure itself, you've seen it before, a little bit more detail. One is called the light chain, and there are 2 light chains. The reason why it's light, it's shorter, okay? And so it has less mass. And if you have a light chain, you must have a heavy chain. It is longer. You can see the structure of this is predominantly beta sheet. And then there's some random coil in there. There's actually no alpha helix. Papain, I said, broke it apart into the FAB region and the FC region, and it will actually digest right here on the heavy chains and break them apart. Now, they're bundles. And do you see in the light chain, there are 2 bundles, right? And on the heavy chain, there's 1, 2, 3, 4 bundles, all right? The bundles get named, and C means constant, and L means light. So that second bundle is the constant light. Now, what does it mean, constant? If I take a look at all antibodies, the amino acid sequence of that bundle is almost identical to every antibody, okay, in the light chains. And then what do you think CH1, CH2, CH3 mean? Constant, heavy, because that's on the heavy chain, 1, 2, and 3 bundle. Okay? And again, those are relatively the same between all of these antibodies, and they would be the same if they're all IgGs. Okay? Then we've got V. C is constant, V is what? Variable. All right. Variable means that there is, when I look at the amino acid sequence, the primary sequence between antibodies, this is where the differences are. It is in these. This is where the binding actually occurs, and each antibody binds very specifically to something. So the ability for the binding to occur happens right at that area, and what it does is it actually happens right here at this interface between the 2 bundles. Okay, the disulfide bonds hold the chains together to make it one working structure. There's a heavy to heavy and light to heavy. And here we've got what are called the CDRs, the complementary determining regions. I might have that typed out on the next slide or so. Complementary determining regions. It's a complement to the antigen. It makes a 3-dimensional structure that will plug in and bind directly to the antigen. Now, I said the variable chains have changes in their amino acid sequence. Well, that's kind of a misnomer. All right, it isn't totally different. Okay, there are variable regions, and if you take a look at it, there are 3 what are called hypervariable regions. The rest of the amino acid sequence, the 1 through 109, probably about 80 to 85 amino acids of those are identical between all antibodies. But there are some highly variable regions that are totally different between antibody and antibody. Now let's see where they are. The hypervariable regions are in red. Okay? And so if I told you the antibody was like this, 2 hands, what really happens is these 3 loops are the fingers, okay, of the antibody. Now, this is a really weird analogy, but these fingers, every one of them, will be different shaped based upon that variable set of amino acids, meaning that have different amino acid side chains there that will make very different 3-dimensional structures. But the rest of this, the beta sheets, all of this part here, doesn't need to really change because this is what keeps the structure of the fold, and so it doesn't matter what antibody are. This part of the amino acid sequence is pretty constant. It's only when you get here, here, and here, or the edge of that variable region, where I need to make the change. Okay? Also, there's some flexibility here, meaning that those beta sheets can actually move around just a little bit. So not only is it variable at the top, but you can also have some flexibility and motion to create unique binding sites for whatever antigen that you're going to bind to. Now, the complementary determining region, or the pocket that actually binds to your antigen, is created between 2 combinations, 1 variable heavy and 1 variable light. And those regions here, those 3 loops, are right here. And so what happens is, this is the pocket between the 2 that is unique in shape. That's what's going to bind. Now, within your genome, there are hundreds of variable light chains and hundreds of variable heavy chains to create millions of combinations of binding pockets. Okay? And so, with that genetics, your own genetics, you have unique ways to create binding variables to bind to lots and lots of things. In actuality, when you are born, okay, you have a whole lot more genetic variability than when you're older. Because there's a process as your immune system develops, you are going to kill off cells that have certain binding pockets. Because what happens is, initially, you're going to have lots and lots of binding pockets, and if it binds to you, that self, we don't want that cell. And so that cell gets destroyed. But other cells that don't bind to you proliferate. And so you get rid of all of those cells that bind to yourself, and then you end up with a host of cells that are available in your body that can bind anything else that might get into your body. Now, we do have diseases where your body starts to react in an immune response to yourself, and those are very hard to deal with. It is where your immune system starts to recognize self and causes the processes to fight off infections or foreign entities, and those are bad diseases to treat. And that's where the antigenic binding domain actually occurs. Now, one thing about the antibody being that little Y shape, remember there's something called dissociation, And so when an antibody binds to something, the binding is dynamic. It binds and lets go, and binds and lets go, and binds and lets go. The fact that antibodies have two binding spots increases the antibody's binding effectiveness. Because if I'm holding on and I let go one, usually I'm still bound to the other. And then it will be dynamic, so it'll rebind, and then this one might go off and rebind. But what doesn't happen, or what happens very rarely, is that both of them let go at the same time for that duration. Even if both of them let go, if it doesn't drift too far, it can bind right back. But this binding is very tight because of that two binding pocket, or the two binding pockets in the same molecule. For immunity to be effective, you must, one, bind very specifically, have the shape perfectly in line with what you want to bind to, and bind very, very tightly. Once an antibody binds to something, that's an indication that it wasn't supposed to be there. And you want to bind to it and flag it so that it is destroyed, whatever it binds to. There are 5 classes of immunoglobulins. IgG is the majority, about 85% of all antibodies are IgG form. They are the principal antibody in serum, okay, in your bloodstream, and they are intended to bind anything that gets in the bloodstream that's not supposed to be there. IgMs are the first to be bound or to be created. Okay? It is not a little Y shape. It actually is 5 little Y shapes cooked together. All right? Here's the reason why. For an antigen to be taken care of, let's say, it needs not to have one IgG bound to it, but it needs to be covered with IgGs. Okay? One binding doesn't elicit an immune response, but a bunch of antibodies bound to it does. When you're first upregulating an antibody, okay, you get these IgMs. It will produce 10 binding sites all in one big mess. If one IgM binds to the entity, it will be tagged to be destroyed. Okay? And so let's say you get sick with a virus. Okay? That meant your body didn't have enough antibodies to bind to that version of that virus to overcome it before you get infected. And so you get a virus and some of your cells get infected, and then it grows more infectious material, and it bursts and it infects others, infects others, infects others. You're sick. Okay? And so the virus reproduces really, really rapidly. Okay? Your antibodies weren't high enough to stop it. So what happens is your antibodies start creating. Okay, IgMs go out first, so that if anything gets touched, it will be destroyed. But you're getting your infection, you're getting your infection, you're getting infection. Then your antibodies then have to start ramping up, and you create lots of antibodies. And what happens is you start destroying the cells because they're infected, and you recover. So how long is your cold? Or how long is your flu? How long do you usually have a flu? A week. Okay, if your immune system is very, very strong, it could be 4 days, 4 to 5 days. If your immune system is not so strong and doesn't respond as quickly, it could be 6 or 7 days. It's all dependent upon you and your immunity, how fast you can get your antibodies up and moving relative to how fast that virus invades. Now, what can you do to boost your immunity if you're heading into flu season? You get a what?
Vaccine.
Yeah, vaccine. You get a flu shot. What does that do? Yeah, what it does is it exposes your body to that version of the virus in a non-virulent form most of the time. Okay? And so your blood sees those shapes. It will start to create antibodies, and so you have a high concentration of those antibodies, in your bloodstream, so when someone sneezes on you and a virus enters into your system, those first few virus particles that get in there, there's enough antibodies to take care of them, and you don't get sick. It isn't that you're not exposed, but you're able to take those entities that are coming in and destroy them before they actually start invading your cells and overtaking. Okay? Yes?
When you create antibodies, if they're not like used within a certain period of time, are they destructive?
Okay, so yes. IgGs last for about 23 days. That's about their half-life. Okay? And they start to degrade. Also, it's a mix between this. Some antibodies that you have, potentially, may represent one cell in your body. Okay? And most of the time, it's roaming around, doing its thing. And if it never touches anything, you'll have one of those roaming around. If by chance it does encounter a virus that it responds to, what will happen is you'll reproduce those cells at the same time as making more antibody. And so you get exponential growth of the number of cells, which then throws out lots of antibodies as I do this race. And usually what will happen is, once you have overcome that, the number of cells go down, but you get what are called memory cells, or a lot more of those. You won't just have one roaming around anymore. You'll have lots of them roaming around. And so that means you become immune to that particular encounter, to that particular virus. Now here's the deal. Some things create wonderful memory cells, and you get lots of them. You can get one vaccination, you're done for life. But many things don't do that. You'll get memory cells and they can last for years, but quite often people have told you, and you may get the advice to get a booster so that you can continue to, good night, you can continue to get those memory cells taken care of. So a lot of vaccinations that you've taken when you were younger, sometimes you do need to get boosters, and quite frankly, a lot of us don't. Unless we are heading to a foreign country on a cross-cultural trip, and you have to get all your shots before you can go. And then, yes, we force that to happen. Okay? Yes?
So, like, after that period, when, like, let's say IgGs last for 23 days,
Mm-hmm.
now after that, is there still IgG in there?
There's still IgG, and you have enough memory cells to be putting that IgG in your bloodstream at a higher level. And then the average level will go down to a basal rate until a stable rate, but it'll be much higher than what you had in the previously or before.
And so if you had the specific like virus that's like has memory cells
Mm-hmm.
too, would that increase the rate of like making more cells?
Mm-hmm.
So instead of like before is new.
Yeah, before it's brand new.
But now the rate would go up.
Yes, you can. Your response, the quickness by which you can overcome it, increases quite a bit. And all right, can you take the flu vaccination and still get the flu? How does that work? Is your immune system not working? Different strains. So, what has to happen is there is a mathematical modeling of all different versions of the cell, of the different viruses that are out there, like H1N1, H1N2, H1N3, H3, 4, whatever. And what the CDC does is they calculate the highest probability for the 3 strands that will probably hit us, right? And they have to order this up way before you get to flu season, so you have enough of the vaccinations. And so they order up a batch, they produce the batch, they give them all out, you get all your shots, and what happens? None of those 3 strains actually hits the U.S. It's 3 different strains that hit the U.S. And so this particular last flu season, there was an A virus and a B virus, of which none of our vaccines actually were created against those. And so people would get the A virus, and then, I've got a flu, I'm not going to get it. And then they get the B, which is totally different. And so some people got it twice, which is nasty. Okay, IgG, IgM, yes?
So since this has 10 antigen binding sites, does that mean it's polyclonal, or is that something different?
No, it is polyclonal also, meaning that, let's see. It's, okay, effectively it's monoclonal. It's only going to bind to one, okay, one spot, but you got 10 places. Polyclonal means that an antigen would have to have at least two different IgGs binding to it in different spots. Your immune system will take a look at that antigen and bind to lots and lots of different places. So when you have that cell produce antibodies, it's not just one, it's multiple cells that are producing antibodies. And so having the polyclonal means that it's binding to multiple antigens. One IgM being produced from one cell will only bind to one epitope, which is monoclonal, but because it has 10 binding pockets, it's enough. Okay. The IgAs, major class of external secretions, so it's in your mucus. It's a first line of defense against bacterial and viral agents. IgE, a very small component, protects against parasites we know, and IgEs tend to be associated to allergic reactions. IgD, at the time that the text that I was using was produced, they weren't quite sure. It is a really, really low concentration class of antibody. We now think it is this. We believe, and this is still a guess. We believe that it is involved in binding to some of our cells that produce antibodies and reducing them, reducing their effectiveness. Why? There are some reasons in which the antigen response, especially on a self-mediated event, doesn't need to happen. These seem to be functioning in a role kind of to reduce response. Again, not quite understood why that would be, but yes.
Reduce our immune
Our
response?
own immune response. Yeah, it'll destroy the immune-producing cells. Yes.
So if that is the case, what do you think that there would be like therapy used for that for like autoimmune diseases?
Yeah, the autoimmune diseases. This is where some people think there might be some ways that we could use these, but it's still tentative. The data is still tentative about how the mechanism actually occurs. All right. So we have 5 different classes: IgG, which is gamma; A, which is alpha; M, which is mu; delta, which is D; and epsilon, which is E. There's also 2 versions of the light chain: a kappa and a lambda. Now, where those differences come in is that constant region. Remember, we said that each had a constant region. The AGMD is the heavy chain constant regions. You get a version of those. And then there's a constant region for the light. There is 2 versions, a kappa and a lambda. What happens when we really do start synthesizing antibodies, the first one is IgM. And it's kind of interesting in that the genome inside the cells, the gene for the M and the gene for the G and the gene for the A are all kind of in line. And what happens is during the life cycle of that antibody producing cell, it first will create a M version, because the M is the first one that is closest to the variable chains. And then what will happen is, as it matures, it will splice out the M version out of the DNA and link it to the G. The actual DNA of that antibody-producing cell changes, and then it will produce the IgG. If it has to go to another, it will actually splice that out of its own genome as it class switches. Okay? All right, let's talk about how this works. All right, so we've got a bunch of stuff in the bloodstream. All right, so you've got those hexagon little things, those are virus particles. Then you've got little pieces of protein that are floating around. Then you have the beta cell, which is the antibody-producing cell, the lymphocyte, or beta lymphocyte, you might see it, and soluble antibodies, and they're all roaming around. Now, if the infected cell releases material, it's in the bloodstream, you can have two different binding events. One, the antibody can bind to the antigen. That's represented right here. So antibody is going to bind to the virus particle. Second thing can happen. The virus particle can bind to the beta cell. Okay? If it binds to the beta cell, two things happen. One, more, so this, one, beta cells are created, and two, antibodies are created. So it will start throwing out antibodies, and it will create more of itself. And so exponentially we'll get a lot of things happen. Here, the antibodies start covering that virus particle. Okay? These virus particles are available and active to bind to your other cells and to infect them. Okay? Having the antibody bind to the virus particle and cover it prevents it physically from invading and binding to your cell and having a cell infected. So covering it physically stops its viral process. But secondly, when it gets bound,
the body's humoral or antibody-mediated immune response begins in the same manner as its cell-mediated response. But here, the macrophages are joined by lymphocytes, called B cells. The pathogen activates only those B cells with matching receptors. These cells stand ready to enter the battle. Meanwhile, the antigen-presenting macrophages activate those helper T cells with matching receptors. These, in turn, rendezvous with activated B cells. Triggered by this meeting, the helpers release chemicals which spur the selected B cells into rapid reproduction. Some B cells become memory cells, ready to respond to a later invasion by the same pathogen. But most become antibody-producing factories, called plasma cells. Freely circulating in the body, antibodies dock with pathogens. This neutralizes them, or marks them for destruction by other weapons in our immune arsenal.
and that's how it sounds inside the cell. Okay? And so, as you can see, the beta cells get upregulated to start reproducing, and then some will start putting out antibodies. Once the antigen is completely surrounded, another cell comes into the battle called the macrophage. Now, what the macrophage is going to do is going to bind to the constant regions of those antibodies. And what it's going to do is say, hey, I've got something. Okay? Antibodies are bound to something that is bad. Okay? I'm going to take it inside of me through phagocytosis. Now, usually, when a virus particle goes inside the body, it's going to infect it. It's going to invade, and it's going to integrate its DNA into the DNA of the cell. The cell is now infected. It's bad. Okay? But a macrophage does it differently. It will bind to the antibodies. It will incorporate the virus particle in a compartment, and then it will fuse it to a lysosome to break it down. And once the DNA and the proteins are broken down, the information is broken down, and it is not infectious anymore. So, the virus particle can get inside your cells two ways. On its own, it's going to infect and corrupt your cell. But if it's ingested by the macrophage, it's going to be brought in and broken down and taken care of. Okay? Here's the second way that I can use antibodies to destroy something. If it's a small little particle, okay, then I can ingest it in the macrophage and break it down. But if it's an entire cell or something bigger, a macrophage can't ingest it. Okay? But an antibody is binding to the surface of a cell that is infected. I need to destroy that cell. The best way to destroy a cell is to break up the membrane. So what we're going to do is called the classical pathway or complement fixation. All right, I'm going to show you the animation. All right, a big protein comes in and binds to the antibodies. And then a whole series of proteins will work in concert. And all of them have C because they're the complementary. And as you can see, as some of them get changed, they change shape and they change function. Many of them break down pieces. And then those broken down pieces will bind to others. But here is the important part. The last stage, C9, there is a series of 9 different proteins. Each one has to work in concert with the next in a series of events. And the C9 creates what's called a membrane attacking complex, the MAC. Okay? The first protein called C1 starts the whole event. C1 binds to the antibodies. C1 is only active when antibodies are there. And then it will process and break down certain proteins. They will bind and create functional groups. Those will process other proteins that'll bind and create functional groups. And then it will activate C9. C9 is inserted into the membrane. Now, one C9 inserted in the membrane is not bad, but it'll insert another one, it'll insert another one, it'll insert another one, creating big holes in the membrane. And as I create more and more holes in the membrane loses integrity, the cell breaks apart. When the cell breaks apart, the mechanism of its function stops and the cell is destroyed, allowing things to drift out. Then antibodies can bind to those, the macrophage can ingest those, and I can take care of the cell. Here's the deal with viruses, though. A virus will come in, the flu virus comes in and infects one of your cells. Here's the deal. The body cannot repair that cell. Because what happens is the DNA of the virus gets incorporated into the DNA of your cell. At that point in time, that cell is compromised. The only thing that you can do to that cell, okay, if you don't do anything, that cell is going to create more virus and infect others. So you don't want to leave it alone. The only thing you can do to that cell is destroy that cell before it creates more virus. And that's where your immunity has to work and overcome the illness. Because what it is, you're infecting cells, you're infecting cells, you're infecting cells, you're destroying cells, you're destroying cells, you're destroying cells. And until you overtake the speed in destroying versus the speed in infecting, you start to recover. Okay? The last thing that we're really kind of talking about is T-cell mediated immunity. Okay, so we're going to look at the T cells or how the cellular component works. And what this does is involves presentation on the cell surface of peptides that were within the cell. All right? Here's the deal. How do you know if a cell is infected? We have to have a mechanism. This is where that mechanism occurs. How do I know if the cell is good or if the cell is infected? Okay? What we're going to talk about is the major histocompatibility complex, the MHCs. Right? We didn't actually know about these proteins, until we started doing transplants. Okay? When you do a organ transplant, what do you have to worry about? Rejection. What you have to worry about is that you put someone else's tissue into yours, an organ into your body. And what happens is your body, hey, that's foreign. And if it's foreign, what am I supposed to do? Break it down and break all the tissue down. Okay? So, your heart's giving out. Someone passed away, but allowed their organ to be donated, and you get a new heart. You have new life. Unless your body starts destroying that heart. Right? And so that's a bad thing. So what you have to have is what? You have to have a heart that what? Matches. Okay, what in the world does matching mean? This is what matching means. Okay? There are about 75 different genes in 3 classes, 1, 2, and 3. We'll talk about 2 of these particularly. And what has to happen here is the donor has to have pretty much the same MHCs as you do. Okay? Because this is how you distinguish between yourself and other things. Okay? All right, 2 different classes of T cells, killer T cells and helper T cells. Killer T cells, if they bind to that cell is going to be destroyed. If a helper T cell binds, it's going to create more antibodies. So those B cells or B lymphocytes bind to helper T cells. Remember when I need to make more, and so it's going to produce more antibodies. Okay? The way that these T cells bind is to bind to these MHC proteins. All right, what in the world? Okay. Killer T cells bind to the class 1 MHCs, and helper T cells bind to class 2 MHCs. So that's important. Killer 1, helper 2. Got to know that. They will produce a totally different biochemical pathway. All right. Here's what happens with a class 2. Okay. And so, remember, had a virus particle. Okay. Covered in antibodies. That's exactly what the body wants to do, right? Virus particle floating around in the bloodstream. Antibodies bind to it. That means it prevents it from infecting, but still, it's dangerous. So it drifts over to a macrophage right here, and it gets ingested, right? But it doesn't get ingested to incorporate, it gets ingested to get broken down. And so the foreign particle is ingested and internalized. It will be broken down. All the proteins will be broken down. All the DNA will be broken down. But in this case, the proteins are important. Now, most of the proteins get totally broken down to amino acids. But some of the proteins, a piece of it, gets married to these MHC class 1s. Okay? And what will happen is the macrophage will present on the outside a protein. Okay? At least a piece of it. All right. The piece that is presenting came from a virus protein that is not self, but it had been broken down. So it's saying, hey, take a look at this. This is the bad guy. This is the shape of the bad guy, and it presents it on the outside of the cell. Okay? The MHC protein is a receptor, membrane bound. Right here is where the protein lies, okay? And it's presenting. On the T cell, the helper T cell, there is a receptor, and a CD4 protein. That is important. Okay? So this is the last thing. I'm presenting an antigen. The peptide came from a virus. The helper T cell comes up and the receptor binds to it. Now, it will only bind tightly if that peptide is wrong. Okay? If it is a foreign peptide. If it does, that allows the CD4 to come in and clamp this whole thing down to cause an effect. Okay, it binds to the constant region and it causes an effect. Now the effect for the helper T cell, produce more antibodies. Against what? Against whatever that B cell produced in that antibody. Okay? And it does that by sending interleukins out. Okay. So, we have a few more slides to go. We will also then, I'll give you an overview of kind of what the test is. I've got to pick it up. It's a different test this time as we move through. And I'll give you some overview. There may be a take-home portion to this test.
So this stuff will be on the test.
This stuff will be on the test. You'll be able to get it completely done and it will be on the test. All right? So, we're almost there. Huh?
Will the full test on?
7 enzymes, and
So this is gonna be like 5, 6 hard settings.
no, this is actually still part of 6.
Oh, okay.
And so, yeah, it's the 6. I didn't know if I was going to be able to get to it, but part.
Okay.
And so,
Oh,
Before you go ahead and send.
I might actually have to, but I need to work through all the other stuff. What's cool about
we're gonna. Did you get a message that we're gonna change our time? My daughter just wanted to know about.
So that's why
I missed Wednesday.
I have it lined up. I just can't produce it because I've been producing. When I get to my desk, I'm gonna hit the produce. It'll take about an hour to get up.
I just want to make sure because I've
Yep.
been waiting for it because I wanted to watch it before Friday.
Yeah.
And then I couldn't, and then I wanted to watch it for today. And then
I totally understand.
Okay.
It'll go up. It'll go up this afternoon.
Which it's fine. I just wanted to. I was afraid that I'd come into class today and you'd be building on it.
No,
So-
totally different.
Okay. Yeah.
And I knew it stopped, and I would go to a different thing.
What is a ascorbic acid do for your
Yeah. There is speculation. Really, there's not a lot of benefit of ascorbic acid. It will reduce a cold, maybe or a flu by 1 day. The connection is tenuous about what it might do. It can help in. There's 2 ways in which things can be degraded. One, lysosome, which is hydrolytic, meaning I break down with water. There's also the peroxisome that breaks it down with redox reactions. The ascorbic acid can help with the peroxisome mechanisms,
So
but
does
that's
it
not
just
necessarily.
help break it down quicker so it can
Mhm.
move on. So
And
basically, the rate of digestion.
The
is
rate of
quicker.
digestion can increase, but it doesn't. It's not huge.